Okay, good morning. It's glad that most of the people who registered actually found their way here, and I think there's a few who didn't manage yet, but there's very few, so it's a good success rate so far. I would like to also say a few comments about the course first. This is a fairly sizable effort of the instructors, and they do this, of course, not as professional instructors. They all have their job as scientists, as students or postdocs or senior scientists. So I think they should be acknowledged for putting this extra effort in to communicate what they know and what they work on. I would also like to say that these courses that Ben mentioned, which kind of are now all the way around, in many cities around the world, have led to a very nice community. So there is a number of groups that participate in this, that like Ben mentioned. And I think this has also had the benefit that oftentimes controversial issues that come up in a field are discussed in a very collegial manner. And we actually managed, as this kind of the group of in instructing laboratories, to write a number of papers together which are used to, I think, represent somewhat of a consensus of some issues in the field. One or two of these I will mention as we go along. So I think this course has additional benefits in terms of building a community, people know each other and kind of trust each other to work together and not against each other. So I think this is, for me, is a very exciting outcome of these courses, in addition to communicating knowledge and know-how to scientists. Okay, so why do we focus entirely in this course on data analysis and not on data generation? So this was actually an issue that came up when we were initially looking for funding for these courses from EMBO and other sources. They said, this is not a complete course. If you don't teach also how an instrument works and what samples are prepared, then this is not complete. And initially they turned this course down for funding. We then could convince them that in fact the major source where the major reason why in proteomics in general, but also in this targeting field, experiments go wrong is frequently at the level of the of computational analysis of the data. I think it is many groups are able to generate very good data, very high quality data from a lot of samples, different samples. And this oftentimes fails at the level of data analysis. So this is a relatively old example of this, where John Bergeron and colleagues did a survey, or it's kind of a test, or community test. This was a simple task for data-dependent analysis, and they sent out 20 highly purified proteins at equimolar level. So that's far away from being a proteome. And they sent this to 27 participating groups, and these groups took on the task to identify the proteins in these samples. Of course, they didn't know how many proteins there were. They didn't know which ones. This was a blind test in a way that these groups got a sample, and this sample contained 20 proteins, but they didn't know that. And it turns out that most of the results were actually quite abysmal. There were people who got 0 proteins, right? There were people who found hundreds of proteins they reported. And then this created some discussion. And we suggested at the time, I had to review this paper, and we suggested we should segregate the tasks apart to see where people went wrong. Was it in the sample preparation, data acquisition, or data analysis? And one result of that was that the data that were generated by each lab was sent to a central laboratory, and this laboratory analyzed all the data from all these 27 groups in aggregate with a consistent criteria and consistent pipeline. And it was very clear that then this informatics part was where most groups went wrong. So many groups were actually capable of producing very high quality data, but when they run it through some form of data analysis pipeline, there was something went wrong. So we thought that in this field of targeting and now DAA, we would like to have a course which focuses entirely on these computational data, and that's why we have this course structured that way. We would, of course, assume that many of the, or all of the course participants would have access to some form of a mass spectrometer in their laboratory or in adjacent laboratories or in some facility where the data are being produced, but the knowledge that you acquire here should enable you to take files that come out of these instruments and actually analyze them in a meaningful way. So this was justification for focusing on computation. And now this is a menu that I'm trying to go through in these next 50 minutes or so. I would like to make some general considerations about proteomics in experimental biology, then answer the question where we should invest our resources, higher number of samples or higher number of proteins, if we have this choice. Then we'll talk about how we might need to integrate large-scale data sets as we can generate them with DAA, with biochemical mechanisms to learn something new about biology, and then have a couple of points of caution when we get to that stage. Okay, so we are clearly living in an era as scientists which is different from the era some 20 or 30 years ago, and that's the era of omics, the era of large-scale biology. And this was, I think, the hallmark of this era was is depicted here in this very iconic picture now, where at the time President Clinton and Prime Minister Blair announced the completion of a draft of the human genome sequence. So with this sequence came a press release from the US government, and in this press release there were predictions made, this was in the year 2000, what we would learn or what we could do as biologists with this sequence. They predicted that we could alert patients that they are at risk for certain diseases, predict the course of disease, diagnose disease, and ensure the most effective treatment, and develop new treatments. Of course, these are all predictions that are related to medical issues, but we could, of course, expand these to general experimental biology, and they would basically state that when we abstract a bit, that we would understand fundamentally how biochemical systems work, and we would make to be predictions about its behavior. So this has not really happened. We are still, of course, struggling with any of those, and this now has taken on different names like precision medicine or personalized medicine in the field of medicine, or systems biology in the general experimental biology field. And we're still struggle greatly to make such predictions. I'll come back to this later on in this presentation. But what did happen is that the cost per genome and the throughput per genome dropped fantastically. So this is Moore's law. This is a law derived from computer science, which basically says that every roughly 14 months or so, the speed of computing, the number of transistors on a chip will double. And this is the drop, basically the increase in, let's say, throughput or performance of genomics, which beat Moore's law by a lot. And this is a famous picture from NIH. This stopped here. I will go on. And now we would be able to say that many people claim that they can sequence a genome for below $1,000, a fantastic drop from the first sequence, which was done in 2000, which cost billions. So the consequence of that was that this is really genomics revolution happening. And this was not really due to the fact that we had an initial map of the genome, but due to the fact that people can experiment with cohorts. And so I would say this genomics revolution was fueled in as much as it could be by the list, by the initial genome map, but much more by the fast, accurate, and highly reproducible measurement of thousands of genomes. So because this allows people to look into patterns, to use machine learning, statistical associations like in GWAS analysis, and to learn new biology which is simply not apparent from a particular genome. So we also now, of course, in proteomics, we're always a bit behind the genomics fields in time. And we also witnessed 2 papers which claimed that they reach an endpoint in proteome mapping, and they explicitly used a draft map, a draft of the human proteome, in their title to make the connection or the analogy to the human genome map. So these papers came out a few years ago, and they claimed basically to have mapped the human proteome to completion or near completion. And this was then, viewed or positioned as an endpoint in the field of proteomics in the point of view of generating a map. So we had also started a project to map the proteome, and this is a community effort. We call this the Human Peptide Atlas, or Peptide Atlas in general. And we started in 2002 collecting data from scientists who did DDA analysis, basically generated spectra. And we simply asked them to donate their spectra once they have published a paper, and then otherwise this spectra would just vanish somewhere in a repository. And you can see here that people were extremely compliant. So initially this was off to a slow start, and then these hundreds of millions of fragment ion spectra were being donated, and this was basically the work of Eric Deutsch at the Institute for Systems Biology in Seattle. And then he computed periodically a cumulative coverage of the proteome. That was what he thought the coverage achieved by these hundreds of millions of fragment ion spectra. So now this, I haven't updated this for a while. It's probably now in the range of 1 billion fragment ion spectra that are donated. And now, of course, there's an exchange consortium, the Proteome Exchange Consortium, where all the raw data that people donate go in, and then internally this is distributed for various types of analysis. So when we now combine these public data with the data from these 2 proteome draft papers, we can see how over time, by the addition of additional large data sets, the human proteome coverage developed based on a number of criteria that are admittedly arbitrary. There is no definitive way that we can calculate the number of proteins represented by a data set. This is a principal reason. This is not that people don't know how to do it, but these predictions of proteins quantified or detected by a number of spectra, fragment ion spectra, is an inference model, and this inference model has certain rules. And so these numbers are always dependent on the rules that are underlying the model, and there's different models. So there's no right answer here, but of course there's an answer which can be correct within the model, or there could be an answer which is incorrect within a model in that it accumulates false positive identifications. So we believe that within a model, and this model here that is used is a model of parsimony, so that this parsimony means that it is the least complicated explanation of the number of proteins inferred from these hundreds of millions of spectra from peptides generated that have been assigned to peptides. There's other models one could use, but this is a sensible model because it is a conservative model, and we would assume that this is roughly a correct coverage of the human proteome to date. So what we do see that at the level of peptides, by adding large data sets, the number of peptides identified went up, and then eventually started to roughly saturate. And at the level of proteins, these additional very large data sets actually did not contribute a lot of additional proteins. So if we say this data set has added 4 proteins to roughly 14,000 proteins, that is certainly within the statistical variability allowed by the FTR. And so this is now one of the first time I mentioned this community consensus or community effort. a community effort was done to re-examine these data from these draft papers and to relate them to the publicly known spectra. And what we can see is that these two papers, which claim to have covered the proteome to essentially completion as a draft, did add some additional peptides, added very few proteins. In fact, this is probably within statistical noise, and that in the cumulative number of proteins that have been to date credibly identified in the confines of a parsimonious model is about 14 to 14,500. So what this means is that, and this was arrived at by 3 different working groups that use different parsimonious models. This includes the TPP, this is from us, this includes Max Quant, and it includes the work of Alexei Nesvitsky and also Ron Beavis, who have developed their own pipelines. So we think this is a fairly accurate result, and it tells us several things. First of all, we are quite far away from a complete coverage of the human proteome. There's a number, several thousand proteins are missing. But it looks like by these methods that we are all using, in bottom-up mass spectrometry, we've reached approximately a saturation. So this now led to a project from the Human Proteome Organization, HUPO, to find the missing proteins, and they go about it again in a community effort to find the missing proteins. It's very, very tough going, because I think the issue is not the mass spectrometer, it is not the data analysis tools, it has to do with proteins, where they're expressed and when they're expressed. Some proteins may be only expressed spuriously under some conditions. And of course there is proteins which be the only, I think, blind spot we still have, which is a significant blind spot, is membrane-associated proteins, which tend to be harder to identify than other proteins. So this is about the mapping. And now, I think, going back to the statement I made about genomics, that the map was occurring, and then the revolution came by the massive number of remeasurements. I think we are in this phase in proteomics. So I believe that these maps are high quality, somewhat incomplete, but clearly show us what coverage can be reached in terms of protein measurements. And now I think we will learn a lot of biology from the fast, accurate, and highly reproducible quantification of thousands of proteomes. So the question, of course, how we do this, and we believe that these DIA methods, also generally targeting methods, have the capacity to generate such data with a relatively straightforward experimental effort. So now I would like to position these capabilities here, that we can measure large numbers of proteins in many samples in the general context of experimental biology. And I would like to make two very short comments about that. I will come back to this a little bit later. So I should ask probably, who of the group here would consider themselves biologists? Okay, so this is probably about half. And who would consider themselves analytical chemists or mass spectrometrists? Okay, so it's probably about, again, a third or half. So those who are biologists, usually also probably many of the mass spectrometrists work in a research organization like a university, like ours here, where we have a very large department of biology. And this department of biology here is divided, I shouldn't say divided, but it's organized in specific institutes that have names like genetics, cell biology, biochemistry, structural biology. And these are traditional entities of biological research which have emerged over the last 50 years. And usually the goal of these efforts is to eventually come up with a mechanistic model of a specific process. This may not be a model that resides in a computer, but it usually resides in the last figure of a paper that's published from any of these research groups, where they say we synthesized the findings into a model, and they draw a figure which represents their view of what they learned from the particular study. Proteomics has an increasingly large role to play in this, and the question is usually asked, can we discover a protein that has a specific function? For instance, a growth factor, an oncogene, a factor that's secreted into the supernatant of a cell line that has a certain function, like insulin or human growth factor and so on, which of course were found a long time ago, but along these lines. And so this is where I think how proteomics was used in the early days, still is used a lot today. And the question basically is, can we discover a protein that carries out a specific function? And to do this, the mapping technique, which is discussed, is an excellent technique, because when we can really exhaustively map and identify all the proteins, we will, by implication, also identify new factors that may have not been known before. So we would like, in this view, to discover many proteins as possible, and find the one that matters. The trouble with these approaches comes usually from this last sentence here. We would like to find the protein that matters. So if we, for instance, which is a common experiment, sequence by exhaustive mass spectrometric sequencing a supernatant of a cell, let's say a tumor cell, or blood plasma from a particular patient cohorts, we will sequence a lot of proteins. We maybe compare these inventories between cells that secrete certain activity and cells that do not secrete a certain activity. And then we look which ones is the differences in these proteomes. And oftentimes there is dozens to hundreds of differences, and the very, very difficult task then is to find among all these proteins, identify the one which has a specific activity. And this is done then in laboratories like cell biology or biochemistry laboratories, and this usually takes a lot of time. So then over time, I think the last decade or so, driven by genomics, there's been a kind of different branch of science or different approach has emerged, and we also see this in our department here. So over the last 10, 15 years, whole groups or a whole institute has been added, which is basically focusing on complex systems and using a data-driven approach where we collect a lot of data. So this is of course pioneered by genomics, and transcriptomics, but now increasingly we do measurements in proteomics, metabolomics, and so on. And the assumption here is that we also arrive at models, but these are different types of models. This may not be mechanistic. There may be statistical models where we work mostly with associations. So the question now that we ask here is also a different one. We're not really asking what is the function of a specific protein that we discover in these data sets, but we would ask rather the question, how is the cell or a tissue or a person or a mouse reacting to the absence or change or mutation of a specific factor? So we basically attempt to observe the system that we're studying. This could be a cell line. This could be an organ of an animal or a clinical cohort. We'd like to observe this subject in many, many conditions, and then learn something by computational techniques that are some form of machine learning or statistical associations. And for that, we would need techniques which are fast, and we would need techniques which are able to measure, like transcriptomics, to a large number of samples quite quickly and in a quantitative manner. And we think that the targeting techniques, SR and PRM, and now at a more deep way, SWATH DIA methods, fulfill these criteria. So the basis for these data-driven approaches is no longer a list or an inventory of 1 or 2 samples that we compare, but basically a data matrix. We have very many samples. I will come back to this, what is many samples mean. And when we measure some or many analytes, and we should have a matrix that has each dot represents a protein observed in a particular sample, and where we have the identity and the quantity of this protein confidently identified. And I think this is really what these methods we discover here or discuss here are about, that the goal that we would like to achieve is that we have such data matrices which can then be explored downstream by computational tools that discover patterns, that discover also relationships that we may not be able to uncover if we are taking a biochemical or cell biological point of view, simply from the complexity of the connections and the context. So this is a picture that is not from us. It's from a paper from actually Biognosis, who will also be teaching part of this course. And this shows the illustratively in a picture the advances that have been realized by these DIA methods to generate such data matrices. We see here such a data matrix. This is generated by injecting the same sample. This was a sample from a lysate that was digested, a lysate of a human cell line that was digested with trypsin. And this same sample was injected 24 times into a LC-MS system with the instrument is a Q-exactive mass spectrometer. And it was injected, the same sample was injected into exactly the same LC-MS system. The only variable here is that the LC-MS system was operated in different mode. And in the blue data, the system was operated in DDA mode, shotgun sequencing, and in the red pillars, the system was operated in DIA mode. So they call this, for reasons which they can explain themselves, hyperreaction monitoring, but it is basically DIA, it's basically the equivalent of SWATH mass spectrometry applied in a exactive instrument, a trapping instrument. So what we see immediately is that, first of all, the red bars are higher than the blue bars, even in one experiment. So this would be one, is the accumulation plot, where the number of peptides identified in 1 out of 1 sample, in 3 out of 3 injections, up to 24 out of 24 injections are listed here. And we see that these are stay relatively high up. So there's relatively little attrition, namely relatively few peptides, which are not seen in other injections. This is the typical decay when we do repeat injections by shotgun sequencing, and obviously we understand the reasons for that, and it's that there's a very large number of peptides that are shown to the mass spectrometer, and it has difficulty measuring every peptide in every injection. So this is the matrix that is generated by DIA. This is the matrix generated by DDA, and the operation of the instrument is the only variable. We also see here the CV of these peptides, of the measurements between the DDA and the HRM measurements, favorable for DIA. So it provides fairly accurate quantification. Okay, so why would we use these matrices that we can generate? I mean, this is 24 samples, not a very large number. We'll then talk about where we would like to go. And this is clearly for biomarker studies, control versus disease, genome or proteome-wide association studies, identification of clusters of proteins behaving similarly across conditions. So this might be, for instance, in a clinical case, a cluster of patients which have a similar protein pattern, and they might then have a similar clinical phenotype, for instance, resistance to a drug or sensitivity to a drug. And we would like to use network inference models to learn about how dynamically networks change, proteins change in the context of other proteins in the cell. So this is how we see these DIA methods in the field of experimental biology. We see this in the area of large-scale biology, where we do data-driven approaches, collect a high volume of high-quality data, so that we can use tools that have basically already been developed to some extent in the field of genomics, for instance, transcriptomic or GWAS studies. And we would like to learn then something about the behavior of proteins in the context of the living cell or a patient or individual in general. Now I would like to comment on the dimensions of this matrix. This is an issue that is an important issue for practical importance, because we do not have in the field of proteomics currently the ability, simply for cost reasons, to go extremely deep to exhaustion on very large numbers of samples. We can reach a high depth, as I showed with these saturation curves, when we accumulate a lot of data, like we do fractionation of samples and do multiple injections, then we can reach basically saturation. But that, of course, comes at the cost of time and labor and sample. So we have basically three methods available. In bottom-up proteomics is the shotgun or discovery method, which samples the detected precursor ions one after one in sequential manner. We have targeted acquisition proteomics like SRM or PRM, where we tell the instrument, especially in SRM, which ions to look for, precursor ions in sequences these ions over a period of time, sometimes over the whole retention time of the LC dimension. And we have these DIA methods which try to fragment or segment this window of retention time versus precursor mass into small pixels and sequence every peptide that is present in one of these pixels, so generating convoluted, overlapping, or contaminated fragment ion spectra that we then deconvolute in a credible way through methods which you will learn. So each one of those can be applied to generate data matrices. I already pointed out some of the differences between the data matrix generated by shotgun or discovery proteomics and DIA, but they all have the ability to generate data sets that have a certain quality. DDA is usually used or oftentimes used for relatively few conditions, maybe few to tens of conditions or samples, and to go very deep. For instance, this has been exemplified, I think, at the highest level by the CPTAC consortium, a clinical consortium of the US government, the NCI. SRM, I think, is well known, and DIA is a relatively new approach, but it has the ability to relatively easily measure hundreds of conditions with a relatively consistent quantification, relatively few missing values, as you will experience in this course. So the question is how, one question always comes up, do we, in DIA world, generate sufficient depths? I mean, are there a sufficient number of proteins identified to allow biologically meaningful conclusions? So this has been an ongoing discussion, and the discussion is continuing, even though the number of proteins identified through experimental advances, for instance, the improvement in instruments, has been steadily growing. I think right now it is reasonable to assume that in a high-end, relatively new instrument, if it's a relatively homogeneous sample set, like the extract of a human cell line, routinely 5,000, 6,000, or 7,000 proteins can be quantified in the duration of about 1 hour of acquisition time. So the number has gone up here a bit, and this will continue to increase, I think, through further improvements in instrumentation. But we're still relatively far away from saturation of measuring every protein in a cell. So what is the relationship between the number of proteins and the total protein mass in a cell? If we have the view that we would like to see how a protein or an organism reacts to a perturbation, for instance, a genetic perturbation, we don't necessarily need to measure every protein. It would be nice, but maybe it's not necessarily an absolute requirement. If we, of course, want to find a specific factor that carries out a specific function, we need to be sure that we measure this factor, otherwise we can make no statement. So here we now, in this data matrix world, we would assume that many proteins that we can measure reproducibly are surrogates for the influence or the reaction of a cell to a particular perturbation. For instance, a genetic mutation or an environmental insult or a drug treatment. And the question then is, how many proteins do we need to measure to get a relatively high level of coverage of these surrogates or these sentinel proteins that we can say we cover a certainly a substantial fraction of the proteome when we do these measurements. So this is a graph that we once generated based on data that were where we tried. This was work from Martin Beck, who was a postdoc here, when he tried to estimate the quantities of as many proteins as he could from a human cell line. Now, these are not super precise numbers, but I think they give you an idea where we are in this discussion. So the x-axis is the number of proteins, and the y-axis is the fraction of the total of the whole proteome of this cell that is covered by the top 1, the top 500, the top 1,000 proteins, if we were to rank the proteins based on their copy numbers, which were estimated here with probably an accuracy of about within a factor of 2. So this would indicate, if these numbers are somewhat accurate, that with about 1,000 proteins we would reach about 90+% of the total protein mass. If you reach about 2,500 to 3,000 proteins, and if we identify them and quantify them, we would cover about 99% of the total proteome in the cell. So now if we look for a perturbation, let's say a genetic perturbation in these cells, or we knock out a gene, or we treat this cell with a drug, we would assume, and it's reasonable to assume, that if we can quantify consistently and precisely in the range of 3,000 proteins, that where we cover most of the proteome, let's say 99% of the total protein mass, that we would be almost certain to get a fairly good impression how the cell reacts to this perturbation. Even though there's a large number of proteins over here, maybe 7,000, which we would not have identified, and the reason they don't contribute so much to the proteome is because they're present at low levels. So this is a consideration where we are, roughly, if we have this point of view that we watch the reaction of the cell to perturbations, then it is reasonable to assume that if we can cover several thousand proteins, we get a fairly accurate and maybe fairly informative picture on how a biological system reacts to some challenge. Incidentally, a human cell like this HeLa cell or HEX cell has roughly the same number of protein molecules as there are people on Earth, which is a remarkable number, I think, in that these cells, these tiny little cells, have such enormous number of proteins. They are present in a concentration of about 300 to 400 milligrams per milliliter. And each one of our cells manages to maintain this enormous concentration without the proteins crashing out. If those of us who are biologists who stuck up their hands, I think everyone would be hard-pressed to get proteins concentrated to beyond 20 or so milligram before they crash out. So this, to me, this is fantastic numbers, and it is actually largely unknown how the cell manages to keep this enormous density of proteins and to keep them in solution and keep them functional. Okay, so now I would like to answer the question or put us in a scenario that everyone who is running a research group or a project will find themselves in. It's a question of limited resources. We now have discussed these different methods. We have their kind of performance profiles, and we have discussed the distribution of proteins over total protein mass. And now we would like to put ourselves in the condition and ask, if we have, let's say, $1 million to spend, and this buys us a certain number of LC-MS analysis, could be also transcriptomic analysis, for, let's say, a biomarker study, how should we spend that money? Should we take a relatively small cohort, deeply fractionate it, and sequence as many proteins as possible? Or should we accept that we can go somewhat less deep, however remain consistent, and go over a larger cohort? So clearly, the CPTAC consortium chose the first path. They spent tens, if not hundreds of millions, to develop a platform to go very deep down into clinical proteomes. And they published a number of really high impact and excellent papers, however, at a very high cost. And we would now like to know, should we go for more subjects or more proteins if we have to make a choice? Of course, we would all like to say we can do everything, but we can't, at least not within this $1 million budget. So this is now, I'm now showing a few slides from Ting Huang, who is a PhD student from Olka Vitek, and in working together, she has analyzed this question as a statistician, and she built models to simulate this question, or simulate the situation to come up with some recommendations. So the way she did this, she took data sets that are published, or not yet published, but generated, and this would be a TCGA CRC, this colorectal cancer study, with spectral counting as the quantification in DDA mode, a colorectal cancer plasma sample data set that was generated by Silvia Surinova here, and she measured this by SRM, and a DIA SWATH data set where we measured 60 cells, cell lines, NCI 60 cells in duplicate, and generated a data set of 120 proteomes by SWATH spectrometry. She then listed she then created for each one of these data sets a plot where she listed the proteins that were identified and quantified in the order of decreasing abundance, or signal intensity rather, and then each dot here is a protein, and then she fit this curve through, which basically indicates the kind of here she shows the standard, list the standard deviation, basically the variability of the proteins. And we see in several, not in this one, but we see in this data set, to some extent in this data set, we see that the lower the signal to noise becomes, then the more variable the measurements become. This is a characteristic of most analytical techniques. If you have a gigantic signal to noise, you can do the measurements very precise. If you like twofold above signal to noise, things become very imprecise and the variability goes up. So this is basically what's shown here. This is DIA data, and this is SRM data where this does not really occur. And this is due to the fact that the proteins in SRM are selected to be nicely behaving proteins, because you spend so much effort to focus on pre-selected proteins. You try to get the nice players. And this was in the spectral counting data where it goes up, and then the variability goes down, and we think this downward trend here is an artifact when the spectral counting gets stochastic. You have either 1 or 0 or 1 or 2 counts. And this, of course, is not a reliable statistical number. I mean, if you have for one peptide 2 spectral counts in one sample, and for the same peptide in another sample 1 spectral count, you cannot construe this as an accurate quantitative difference. Okay, anyway, that's what she did. She did this also for different data, and I would like to just point out this one here. And this is data from a Lumos instrument, a trapping instrument. And this is really beautiful data in the sense that over a quite large dynamic range, we see also this trumpet plot behavior, but it is very much attenuated. So for now, we would say that these trapping instruments operated in DIA mode, produce the nicest data that we can generate from the point of view of quantitative accuracy and dynamic range. Although we cannot directly compare this one, for instance, this one here, because what we show here as variability is the cumulative technical and biological variability, and here we analyzed different sets of stem cells which are very, very similar to each other. So the biological variability is small, and here the NCI 60 data set, sample set has samples which are very different cells, so there is a much larger amount of biological variability. But in any case, we see that Ting generated these plots and basically learns for a specific type of experiment the noise behavior of the proteins in that type of experiment, and this then she uses to do simulations. And she does the following simulations. These are these 5 quintiles, which is now represented here by the SWATH NCI60 data set, and she basically shows then the proteins in each quintile. So this would be the highest expressed proteins, and these would be the lowest expressed proteins, which have the highest variability. And she would, in a computer, put in in each one of these quintiles, 3 biomarkers. And these are synthetic biomarkers, so they're not real proteins, they're simply simulated in there. And the biomarker would be defined as a protein that is 1.5 times as different in abundance between a case and a control. For instance, a disease case and a healthy individual. It's a very typical assumption for biomarker discovery tools that you have in a lot of other proteins, some proteins which change their abundance and therefore are indicative of the predictive of the class the person belongs to, patient or control. So this is the experimental setup, and she did a lot of simulations. She simulated if these data sets consisted of 60, 500, up to 5,000 proteins, if this data set consisted of 10 to several hundred, 400 patients or samples, and then she predicted or simulated whether the distribution of predicted proteins was all over the abundance range, more in the high level or more in the low level of proteins. So she did hundreds of simulations in a computer, and this is basically what comes out. So this is to some extent intuitive, to some extent it's non-intuitive, but I think very informative. So here she shows, we have these plots here. This is the number of proteins increasing. So this would be the more proteins we measure and factor into the analysis. This would be the number of subjects increasing from few to several hundred. And then she shows here the accuracy of the prediction in a biomarker experiment. And this would be the number of correctly classified subjects, true positive or true negative, over all the subjects. So we would have like to be here at 100% or a number 1. This would mean it's perfectly accurate. So what we see here is that when we have relatively few samples in a cohort, we're not doing so well. And interestingly enough, the more proteins we add, the less well we're doing. This is also true when we increase the number of subjects. We're always doing not as well when we keep adding proteins. And this is, of course, quite nicely explicable by the fact that if we keep adding proteins, we will naturally add proteins which are noisy, have a large standard deviation. And so we basically, if you're able to identify biomarkers up here or up here in these proteins which have very good signal to noise and a low variability, if we add a lot of proteins that are highly variable, simply because we're getting down to the signal to noise, which is a property of every analytical technique, we drown out or dilute out the signals by noise. So this is clearly what comes out of these simulations. So clearly, every time we make the cohorts larger, we gain accuracy, and we see here that when we are in the range of 300 to 400 subjects, and we have a reasonably high number of proteins, we reach a very high level of accuracy. And the same trend is observable when we ask what is the sensitivity of identifying predictive proteins, really biomarker panels. We see that we're not doing well with low number of cohorts. We do very well with high number of cohorts, and every time when we add more proteins, we need also a higher number of samples to get to high predictive sensitivity and predictive accuracy. So the lesson of that, she did this she did this also with the different data sets. This is the DIA data set. She did it also for SRM, for DDA, and for the Lumos data set. And basically, they all show the same trend. So the conclusion we should draw from this, I think, is important for the field of proteomics, what we're discussing here from a technical point of view, that if you are able, first I should say, this does not just apply to biomarker discovery experiments. This is a general trend. This also applies to machine learning, also applies to unsupervised clustering. It is simply the fact that when we want to gain confidence, here shown in the predictive accuracy, or the consistency of identifying the separating proteins, the proteins separating the groups, we'd rather spend our million on generating a larger number of samples, rather spending lower number of samples and go very deep down. So this is a very convincing conclusion from all these simulations. And when we look in the proteomics literature, there's very few studies published that are in this range of 200, 400, or even 1,000 samples. And I think that's where we need to get to be serious players in this big data or data-driven arena where transcriptomics has been so successful. Many biomarker studies have 10 to 20 sample cohorts, and we can already use this type of table or simulation that we could conclude that these biomarkers discovered or hypothesized as biomarkers, these proteins, are very likely to be unstable and maybe not turn up to be validated. So this is the conclusions from this part. More proteins decrease predictive accuracy and sensitivity of identifying predictive proteins. More subjects always in every scenario with every technique, it increased the accuracy and sensitivity. Less technical variability increases performance. And the conclusion is that if we want to further develop the mass spectrometry-based proteomic field, we should invest in optimizing the throughput of biological samples and reducing the technical variability over the number of proteins identified. I'm not saying we should not also, of course, get deeper down, but for this data-driven approach, it is very clear from the simulations that the higher number of samples always beats higher number of proteins. So now let me go into this, and then for about, do you have 15 minutes more?
Yeah,
okay.
We have 25 minutes.
Oh, okay, good. So I thought I was running out of time. So now I discussed the fact, or at least the opinion, that with this DIA technique, we are entering or trying to operate in the data-driven world where we operate with these data matrices. And I try to show that we should invest in getting data matrices with a large number of samples because they are more informative than data matrices with few number of samples, even if we can go very deep down. Now, the question, of course, comes up. If we are biologists, is this enough to explain biochemical mechanisms? This is what we really would like to know. We would like to learn new biology. And for those who follow genomics, you've probably seen that there has been genome-wide association studies that now include hundreds of thousands of individuals or genotypes where genomic variants are being sought that would associate or correlate with a complex disease like cancer or diabetes, or for that matter, a complex phenotype like body size. And this has been largely ineffectual. It has been extremely effectual even from the point of view of finding factors, genomic loci, that associate in somehow involved in creating this phenotype. For instance, tall or large body size, or a propensity for a person to develop diabetes. But from a mechanistic point of view, how these genes or mutations thereof contribute to this complex disease, this has not been really very interesting, or at least not very high yield. So connecting these associations to biochemical mechanisms remains a substantial challenge. And now if we want to operate with proteomics in this big data world, we should ask how do we position this proteomic data in the best possible way, that we can learn something about mechanisms, and that's the topic of this discussion. Everyone who writes these papers in proteomics, including us, we always will say as a justification of measuring proteins as opposed to transcripts, that proteins are closer to the function than transcripts. This is certainly true, but in some level it's a bogus argument, because if we still do not find a way to link the proteins to the mechanism, even though they're closer, we still are not where we would like to be. So this is now what I'm trying to discuss in the next few minutes. So a lot of this large-scale or big data analysis or world is based on correlations, or more generally, associations where we have two dependent variables, so they have something to do with each other, a gene mutation or an abundance of a transcript with a body size, for instance. So, and we would plot then a number of individuals or measuring points, and then we draw a curve, and if it's the curve, we draw a curve, we would say these two variables, x and y, are positively correlated. For instance, the more someone eats, the heavier that person becomes. It's an association. It doesn't tell us anything how this functions biochemically. So this does not imply a causal relationship. It's simply the coincidental observation of two variables. So this has then led to people, scientists, computer scientists, who to predict in this article, which is now about 10 years old, the end of theory, and they said the data deluge makes the scientific method obsolete. So what they mean with the scientific method is that we learn as scientists to read papers, to formulate a question or hypothesis out of what we learn, and then to design an experiment that tests this hypothesis. They say this part of research is now obsolete. We don't need this anymore. And this is text they wrote there and said scientists are trained to recognize that correlation is not causation, that no conclusion should be drawn simply on the basis of correlation between x and y. Instead, you must understand the underlying mechanism that connected to, once you have a model, you can connect the data set with confidence. Data without a model is just noise. So this is clearly what we're trained to do. And what they say, in fact, with massive data that we have now available, increasingly also in proteomics, this approach, hypothesize, model, test, is becoming obsolete. They say correlation is enough if we have large amounts of data. So this, of course, is an interesting proposition, and because this would mean that if we do, if we look at the biological research institution, like a department like ours, this part of the picture we could basically eliminate. We would say, who cares? We don't need biochemists. We don't need cell biologists. We don't need structural biologists. We just do a lot of measurements, preferably, or maybe proteomic measurements, and we use the power of computers. This is what they predict, and this has caused us to think about this in fairly interesting ways and to actually test this prediction. I would just like to say now, I do not believe in that, but that's not really relevant. But I show now some data where I think we illustrate, this is not a proof, of course, it's just a case study, that correlation is not enough, and that correlation, together with biochemical mechanistic understanding, actually is probably the most powerful approach to learn new biology. So this is what I would like to now show in a few minutes. So the study goal was to link precise quantitative proteome measurements to genomic and phenotypic variation in a genomically well-defined system. So this is basically what we would like to do in personalized medicine or precision medicine. we have genetically well-defined system. In this case, would be humans, for whom the genome has been sequenced. We would have a phenotypic measurement of these humans, and for instance, they would have a certain disease that we would like to know, does this person respond with that disease, respond to that treatment or treatment B, or treatment A, or some other thing, some other treatment. So we like to go from data to mechanism. So the system we study is not humans, because there the genomic variability is enormous, but there's a system of, or cohort of mice, which we have access to through, provided very generously through a consortium, particularly Johann Overks, who is a professor at EPFL in Lausanne, who is part of the group of scientists who created this resource. So this is referred to as the BXD family of mice, and it is currently the largest and best characterized mouse genetic reference population. So what is a mouse or a genetic reference population? It is a cohort of strains, in this case about 180 strains, and each animal in each strain is identical to the other animal in this strain, at least genetically, but the strains are different between each other, and they are different genomically, and they're different in a certain structured way. So you could, of course, go and collect mice outside, wild-type mice, and they would also be genetically different, but the difference would be more of like a human population, a very complicated pattern. So these have simplified patterns of variability, and this arises from the fact that two mice, a C57 and a DBA mouse, were initially crossed. Both of them are very well characterized laboratory mice. And then through two generation crossings and then out crossings of around 20 generations, 180 strains were generated, which have the property that they are different amongst each other, but the differences are not new alleles. For each locus, you have only two alleles, one from the black 6 and one from the C57, and the difference that we see here is simply reshuffling, a redistribution in 180 different ways of the alleles, of the loci that were present in these initial mice. So they have a very structured, so basically for those who are not interested in genetics, these are like brothers and sisters in a family, except you have 180 of them. And we have from each brother and sister, we have basically clones. So this is the resource. And then these animals were phenotyped through hundreds of phenotypes. For instance, how much they breathe oxygen, how much they run at night, how fat they get when they eat fat food, how old they get if under certain conditions, and so on. So it's a fantastic resource because we can relate genomic variability to many phenotypes, many of which are clinically relevant. So Yibo Wu and Evan Williams worked on that in our group, and in the first study, they selected 192 proteins that are relevant for metabolism and measured them across a subset of these strains, 40 strains, where the mice were grown either in normal food or in high-fat diet. Some get very fat and get diabetes, others don't care if they eat fat food. So this was the experiment. So we have two axes. One is the genotypic axis, that these 40 strains are genotypically different. We have a environmental axis, which is diet, and then 190 proteins and transcripts were measured from each one of the livers of these mice. The proteins were measured by triple quadrupole mass spectrometry and in SRM mode, and the transcripts were measured by a gene chip. Then we also remeasured this by SWATH mass spectrometry, but it's really not relevant for this discussion here. And then these patterns emerging were put in relation by association studies with phenotypes like liver weight, body weight, metabolites, lifespan, and so on. So Yibo generated 3 replicates, technical replicates, 3 biological replicates of these measurements. And this is the overall CV of the technical replicate. So these are highly reproducible data with less than 10% CV, which is for proteomics very good. And this is the data set that emerged. We see here the CV of the peptides is about 30%, which clearly means it is worse than the technical replicate, which means we see biological signal. And then this was the matrix. We see it has very few missing values. It's 192 proteins in 77 mice cohorts. We can now do associations. We can associate the genetic variability, namely the allele that's present in each locus of the mouse genome, with the abundance of a protein. And so this is referred to as QTL, quantitative trace locus mapping. And when we do this with proteins, this is called pQTL, protein QTL. It basically establishes a statistical relationship between a locus present at an allele present in whether it's the black 6 or the DBA form of the gene at a particular locus, and the abundance of a specific protein. So we identified 30 of those which are acting on themselves, and 14 act on some other protein that's not coded for by this QTL locus, and some are dependent on the food. So from 190 proteins is actually quite a good yield. So this would be total 44 QTL. So we find 44 genes that affect directly or indirectly the abundance of a particular protein through genetic associations. And so this is now what we could say. We could say this is basically what genome-wide associations are doing. Of course, they do it with many more genes, actually millions of SNPs. But that's basically what they do. They establish a relationship between a genetic variant and a measurable trait. For instance, a phenotype in our cases would be a protein abundance. So now we wanted to see whether we could now test this hypothesis that bringing in prior mechanistic information would teach us something about the system that is not apparent simply from associations. And this is one example of this where I think is quite an interesting example. One of these QTLs, of these proteins, for which the abundance is controlled in ways we don't know mechanistically how, but it is strongly correlated, that particular locus on that gene, with the high or low level of abundance of this protein, is an enzyme that is the last enzyme in the degradation of branched chain amino acids, which is a biochemical degradation pathway, which is as usual goes through various steps of sequential enzymes. So this we know from biochemistry, and that's also how these proteins were selected to do the SRM measurements. We also know what these molecules are, the intermediate products that are being generated when, for instance, the amino acid lysine is degraded to basically completely degraded into small molecules. One of them, so they have names, and one of them is amino adipate. And so we can measure by proteomics basically the abundance of this protein, the enzyme. We know its relationship to the genome. So the genome drives its abundance. We can measure its abundance. And we can now test the effect of the abundance of this protein, presumably its activity, on the concentration of these intermediate products, small molecules, degradation products, in the cell. So this was done by Yibo, Wu, and Evan, and they show two correlation plots, as I showed before schematically. So they show this first one correlates the amount of the enzyme present in the liver of these mice with the amount of 2-amino adipate, which is this intermediate product. And we see the more of the enzyme is present, the less of the 2-amino adipate, the metabolic intermediate product is present. So what that means is that there is a certain flux through this system. If there's a lot of activity down here, this is like a water hose. The water hose goes fast, and nothing accumulates up here. So the intermediate products are basically processed all the way through. If the amount of the enzyme and its activity is low, then these intermediate products pile up. So we can say this simply from this correlation, and now we have made a link, a mechanistic link, from a genetic locus, which determines through this pQTL the abundance of this enzyme, to the concentration of an intermediate small molecule metabolite. This certainly could now be fit into a mathematical model because it's mechanistically understood how this works. This is simply to show that two intermediate products correlate positively with each other. That means if the water hose is closed, both of them pile up as we would expect. So now this is not by itself super interesting, but we can now again use this mechanistic insight into look in the literature whether these intermediate metabolites, which have been studied extensively by metabolomics, have an association with disease, and they do. So 2-amino adipate, which is this intermediate product, as we have established, the abundance of which is correlated by a genetic variant in these mice through the expression level of the protein, the enzyme that controls the flux through the system. This particular molecule was found as a biomarker for diabetes from a very, very large study, the Framingham Heart Study, which has used genome-wide association metabolomic analysis to establish this link. So what we can claim is that through the combination of relatively large-scale proteomics, I mean, this is still not, of course, very large scale, 80 samples versus 192 proteins, but it goes in that direction. We can work with patterns, has established together with statistical analysis like GWAS analysis and a mechanistic link that explains how this particular biomarker for diabetes risk is controlled genetically, at least in these mice, and further worked and showed that this mechanism is actually conserved in humans. So it allowed us to do a link between large-scale association studies where tens of thousands of people were genotyped, and to link the biochemical knowledge and some insights created from moderately large proteomic studies to propose a mechanism that links genomic variability to enzyme abundance and enzyme kinetics, and to eventually a biomarker for disease link, explaining mechanistically how this particular marker is actually controlled. So we conclude that correlation is not enough. This is certainly the mode of operation we use in our group. We would want to generate large-scale data sets by DIA measurements on large cohorts, as I described before. But we would like to see whether we would be able to incorporate in such models biochemical knowledge from pathways and biochemical regulatory systems which have been established before. And so I think the question is, how do we best use the prior information into which millions of research dollars and research papers have been established into, and how do we bring this information into the big data world? I think this is an extremely interesting challenge. I think proteomics has a lot to offer there. Now, so finish.
No, I think
it's fine. Okay.
7 or 8 minutes.
Anyway, this is very short. I would like to have tale of caution. So we have now established that it is beneficial to do very large cohorts, if this is possible. And I try to explain how we think the data from such cohort measurements by DIA mass spectrometry, or for that matter any other method, are beneficial to on how they can be positioned in the field of experimental biology. So whenever we go to large sample numbers, we are now facing, we learned this the hard way, we are facing new problems that need to be addressed. So doing a cohort of 1,000 samples is not the same as doing 100 times a cohort of 10 samples. A whole range of new problems come up, two of which I will briefly touch on, and then they will be certainly explained in more detail across through the course. But we cannot just, the main message is we cannot just do what we have been doing for 10 to 50 or so samples and just say, okay, now we run 100 or hundreds of samples or thousands of samples through the machine because new problems creep up that we need to be careful to address, otherwise we might be misled. Okay, the first one is the accumulation of false positives. So this is true for DDA analysis, this is true for DIA analysis, and probably for any form of mass spectrometry. So the problem is the following. Here we show, this was initially addressed by Manfred Claassen, who was a PhD student here, has now his own group. And it was in response to findings from this from the Peptide Atlas project that the more proteins, the more spectra were added to the database, the more proteins were identified. So this made us suspicious, and then because there seemed no end in sight. And so I will now explain what Manfred Claassen did here, at least what the underlying assumption is, and what we should pay attention to if we accumulate large data sets in a cohort study. So the dots here are proteins of the human proteome. Presumably, this would be some 20,000. When we now run a typical DDA experiment of one sample, we will run these samples through these peptides. We run the peptides through a database search engine. It uses presumably some form of assignment control, assigning some scores, calculate some FDR, for instance, with a target decoy model, or in some other way, and we identify a number of peptides which we believe are correctly identified, and we identify a number of peptides, or we designate a number of peptides which are false positives. Let's say if we have a 1% FDR, 1 in 100 would be wrong. We don't know which one, but this is what statistics would tell us. So this is pretty good because we have this well under control. Now the observation was that when we keep adding data sets, so for instance we do the same thing 100 times, inject the same sample 100 times into the instrument, and in each run we have a 1% FDR control. And so we observed, or Manfred observed here, that the peptides which are truly identified, the true positives, will accumulate, basically pile up on a number of proteins which we already knew were present. And the red ones, which are the false positive 1%, they have no reason to accumulate or aggregate around these proteins. They have an equal likelihood to hit on any protein of the human proteome because they're wrong. There's no relation between the spectrum and any protein because they're wrong assignments. And the wrong assignments have equal likelihood to be from any one of these proteins. So if we do this long enough, if we keep adding data, we control in each run the FDR, let's say at 1%, but we don't have a global FDR control. We observe that eventually we have hundreds or even thousands of identifications of these proteins, which are undoubtedly present in the sample. And we have an accumulation of red ones, essentially hitting on every other protein because this would be the 1% false peptides, and they will basically eventually hit every protein. So when we graph this, if we keep adding number of peptide to spectrum matches, we would assume that when we, let's say, inject the same sample 100 times or 1,000 times, we would assume that eventually we have identified every protein correctly, and then these protein numbers, which is the y-axis, should saturate out. And if we control the overall data cohort false positive rate correctly, we should eventually reach saturation. If we don't control the overall cohort FDR correctly, we see the black curve, where the more data we add, the more proteins we claim to have identified. And the delta here, so this is a sure tell sign that something is problematic. If we keep adding data from roughly the same samples and the number of proteins keeps creeping up, this is a sure sign that something is wrong with the FDR control over the cohort, not in each particular run. And because eventually it should saturate out. And the delta here between the green and the black is essentially accumulation of false positives. We know now, I talked about these two proteome map papers at the beginning. We now know that this is exactly what happened in these papers, and they did not consider accurately enough this accumulation error, and therefore came up with vastly inflated numbers of proteins that were claimed. The same exact thing happens in DIA. This was actually to some extent a surprise, and George Rosenberger, who is here, as a community effort from a number of groups that are active in this field, found, together with Isabel Bludau, who is also here and an instructor, they found a way to control and understand and control the FDR accumulation for large DIA cohorts. I don't want to spend a lot of time on this graph because I'm sure George or Isabel will talk about it. But this basically depends on the size and the type of the library, and here we simply have additional runs, the number of runs increasing, and we see without proper control we have an increase of number of proteins which are claimed to be identified, which is exactly what happens if we don't do proper control over the whole aggregate of the experiment or the cohort and focus the control only on each run. This could then be controlled through mechanisms that George will certainly explain, and we see here nicely curves, which then the more samples we add, doesn't really matter. The number of proteins identified remains stable. That, of course, only applies if substantially similar samples have been run. So this was a case where we had 230 runs of identical sample injected to same sample injected 230 times, and this should eventually lead to saturation, and it does. So this is one tale of caution, that what we learned as doing FDR control, or basically quality control of the data, what applies to one or a few runs does not necessarily apply to hundreds of runs, and we need to have new devices. And I would claim that through this community effort in the world of DIA, this is fairly well understood and fairly well controlled, and you will learn these procedures. The second tale of caution is batch effects, and this is an example from the literature. So this is not in proteomics, but I just want to illustrate the point. This is from genomics, actually, more specifically transcriptomics, and it's from the ENCODE consortium. These are certainly people who know what they're doing. So the question they asked was, if we take a mouse apart into different tissues, and if we take a human body apart into different tissues, and we do transcript measurements from the, let's say, the heart of a mouse and heart of a human, liver of the mouse, liver of the human, and so on. And then they clustered the patterns that were the transcript patterns by unsupervised clustering. And they published a paper in Nature where they said that the human tissues cluster together and the mouse tissues cluster together, but apart from the human ones. So basically what they said, that there is some form of humanness to tissues, to transcript expressed in tissues, which fundamentally is different from the mouseness of transcript profiles expressed in tissues. So this was this paper. Then this data is actually not very plausible for a biologist, and this was re-examined. And then it was found that this was all an artifact. And it was an artifact not because the people didn't know what they were doing. It's not a computational artifact, but it's a batch selection artifact. It's a batch effect that crept in that they did not consider. And what they did here, this is hard to read, but the red are the human samples and blue are the mouse samples. So they analyzed, for the most part, the human samples together in a flow cell of a Illumina sequencing machine. And they basically analyzed the mouse samples together in the flow cell of an Illumina sequencing machine. And what they found, what basically the source of the graph was that I showed is a reflection of a batch effect in the flow cell of the Illumina sequencing machine. So by the fact that all the mouse and the human samples were basically, all the mouse ones were in one batch and they're sequenced through, and all the humans were in one batch and sequenced through, it turned out that the machine probably had a somewhat different performance, and this created differences in the patterns that were bigger than the differences between the different mouse and human samples. So they concluded the wrong thing, and when this was redone, we see that testis, human, mouse clustered together. Same is for brain, spleen. Basically, now the exact opposite conclusion was drawn when the experiment was done properly, and that the tissues, so the same tissue between mouse and human, is more similar than the difference between the species. Completely orthogonal conclusion due to a batch effect that was not considered, a confounding effect that was not considered in the initial experiment. So this was then also published and was an interesting case, and they concluded that the conclusions are unwarranted, they're not wrong. This is on the first paper, because the study design was simply not suitable for addressing the question of tissue versus species clustering of the gene expression data, because the experiment was designed wrong in the sense that confounding factors like the machine performance was not considered and could not be detected simply because this was not present in the experimental design. So the reason I'm bringing this up is that if we need to consider, if we go to large cohorts, possible sources of batch effects, confounding factors, and these could be, like in this case, the performance of the machine. If we do a large cohort and we use 2 or 3 mass spectrometers, we do not want to end up at the end of the study say, well, we found that mass spectrometer 1 and 2 and 3, on which the samples were run, had a different performance profile. And so we should make precautions, and Olga will extensively talk about this, to avoid batch effects and to make the structured experiment such that if they occur, they can never be completely avoided, that we detect them and can, to some extent, correct for them. And this would be, a lot of thought has to be done on the experimental design. And so the one effect is to form blocks of samples and to randomize within these blocks and have overlaps between blocks. So these are things you will learn, but this is a very fundamental problem that in this type of large-scale biology, that we need to assume that things happen that we cannot control and that creep up and have the potential to completely distort our data and conclusions. And so this is something we need to consider, and Olga will talk about that. So with that, I would like to thank you for your attention. I wish you a good course, informative course, and hope you learn a lot. And don't hesitate to ask questions. Everyone will be very happy to answer questions to the best of their knowledge. Thanks a lot.